Okay, hi everyone. This is Mrs. Shannon, and I am going to go ahead and go through the identification in the nutrients lab and how to do the actual individual tests to test for proteins and carbohydrates. So my equipment is out. I'll go through the items as we go through. Notice I have my glasses on. I also have gloves. If you are anywhere around any of the lab tables that have the lab being conducted, even if you're not doing it, you're to be wearing goggles at all time. If you're doing the lab itself, you will also want to wear gloves. Some of the reagents may irritate your skin if you don't do that. So what you're going to need to start with is 3 test tubes. The test tubes are being kept in these little blue racks. It helps keep them upright. So this is the test tube rack when it refers to it in the pre-lab. For part 1, you're going to perform the first test on 3 different substances. The first one will be the energy drink that you're assigned. The second one will be distilled water, and the third one will be a soda. I have Mountain Dew today. I don't know if that's the same soda that I'll have when you do it, but you'll need to use one soda as a comparison. So I'm going to run through these tests just using the actual soda. I'm not going to use the energy drinks or the distilled water at this point. Notice that the distilled water is going to be used as the control. The energy drink is your experimental, and this is another experimental that we can use when we do our comparisons. So to begin with, you are going to obtain the 3 test tubes, and you're going to want to label these. So what I'm going to do is take tape and put on, and you'll probably want to do this before you actually put your gloves on, a little piece of tape, and you're going to number these 1, 2, and 3. Make sure you use Sharpie on the tape or a wax pencil so it doesn't come off. Okay? And so in the first test tube, transfer 5 milliliters of distilled water. The second one, the energy drink. The third one, the soda. So here I go. Just going to pour a little bit of soda into the cup to make it easier to pour into the graduated cylinder. And make sure you find where the 5 is. Remember, when you're using a graduated cylinder, you want to get your head even to where the meniscus is going to be. So I'm bending down. I'm keeping it, on the table. Now what's cool about biology is we do want to get as close to 5 as we can. We are not as exact as chemistry, so if you're just a tiny bit over, under, if you can't tell for sure, you're probably okay. Also, this is a qualitative lab that we're doing, not a quantitative. Even though we are dealing with numbers to set up, we're going to be looking at results that are more along the color spectrum, more along the color line. We are not going to be measuring our resulting data with actual numbers. And so rather than quantitative data, we are going to be collecting qualitative data. So pour your corresponding beverages into the corresponding test tubes. So I'm just going to try it with my first, and you'll have something like this. Okay? Now you're going to want to add 5 drops of burette reagent to the test tube. The burette reagent is going to have the blue cap on them. They will be labeled. They'll also be labeled on the lab table itself where you pick it up, blue equals burette. This is going to test for the presence of proteins. So you're going to add 5 drops of burette reagent to each of the test tubes. So again, I'm just doing my 1. If you get 6, it's okay. 1, 3, 4, 5. And I added a 6th one simply because I knew that I hit the side of the test tube and it was going to slowly go down. And so just to be on the safe side, I went ahead and added it. Now I never point this towards my face, I always point it away. And it says to gently stir it. Okay, it's low enough that I can just kind of swish it around like this and we should be good to go. So gently shake the contents of each test tube. Caution, on the pre-lab it says it's a strong base, so if you splash some on yourself, you want to make sure to wash it off immediately and notify Mrs. Shannon. Okay, if you notice a color change from blue to violet, it will indicate the presence of a protein. And now the liquid that we put it in this case was yellow. We didn't notice any significant color change there, so we would probably mark it as negative. If in doubt, you can always add a couple more drops. It's not that big of a deal. You're going to repeat with the water and the energy drink. You're going to record your results in the data table. At that point, you're going to pour them out into the sink, rinse it out really well, and then prepare them for the next test. Okay, any questions on that? Okay, the second test that you're going to do is to test for simple carbohydrates. A simple carbohydrate would be like a monosaccharide testing for glucose, a monomer of carbohydrates. For this one, what you're going to do is you're going to do it the same way. You're still going to need 3 test tubes for your 3 different substances. Okay, again, just like the previous test tube, you should already have them labeled from before, but you're going to measure out 5 milliliters of the specified drink. Whoops, I can't measure directly in there. So I measure out 5 milliliters. And now to test for simple sugars like glucose, we use a different reagent called Benedict's. Benedict's is an indicator that will turn color in the presence of simple sugars. And so what we're going to do now is we're going to take Benedict's. Now Benedict's will be labeled. It also has a green top, green for Benedict's. And in this case, let's add about 10 drops, and that's what it says on your lab. So, again, away from my face. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So 10 drops are added. Okay. For this one, it says that I'm going to place it then in a test tube for a water bath for 3 to 5 minutes. There should be 2 water baths going on in the classroom. A water bath is simply a beaker of water placed on a hot plate to keep it warm. It shouldn't be boiling. If it is, let your teacher know so they can adjust it. But you might be able to see the water droplets on the side. So I'm going to place this in. Make sure that the end is pointing away from anybody, please, when you are putting it into the water bath. And we're going to let that sit for a few minutes. And so right now it says 3:56 by my computer, and we'll check back in a couple of minutes. While that is going on, I'm going to do the last test. The last test is also a carbohydrate test, but it's going to be a little bit different. Instead of doing simple sugars, we're going to look at complex sugars. Complex sugars occur when you have different monosaccharides coming together to form a polysaccharide. The test that we do for this is going to be an iodine test. Iodine, you may be familiar with in the medicine cabinet. It's often used as an antiseptic to cleanse areas that you might have cut open, or if you have a scratch. But it can also be used as an indicator to test for complex carbohydrates such as starches. I is going to be the iodine test, and it's the yellow cap. So again, for the complex one, I'm going to do 5 milliliters. Okay. So here's my 5 milliliters. Now in this case, what I'm going to do is I'm going to take iodine, and I am going to go ahead and add 5 droplets of this. So we have 1, 2, 3, 4, 5. One of them hit, so I'll add another one. And I will turn it. Now the thing with iodine is that in the presence of a complex carbohydrate, what that will mean is that it will turn a very dark color, almost like a blackish purple. If it stays a nice golden brown color like it is, that means that there are no complex carbohydrates present. Now the difference between complex and simple carbohydrates, they're smaller. It's easier to break down. It's easier to retrieve that energy. So it's going to be quicker energy, not as much work to get out. That's what's going to be used first. After that, then your body's going to dip into the starches. The starches still have a lot of energy in it, but it's going to be a longer molecule, so it's going to store more in there. Starches are often associated with pastas, for instance, and so when we talk pasta loading or carb loading before a big race, for instance, that's why people do it, because they're storing a lot of molecules that have long chains of carbohydrates in them that are going to be able to break down and provide that person with a lot of energy in the long run. So complex carbohydrates are actually a good thing for your body if you're going to be using them. If they're not going to be used, on the other hand, they will be stored in your body and converted over into fats. And that's oftentimes if you're eating a lot of complex carbohydrates and starches and you're not exercising, you'll see that you might be putting on a little bit more weight as it's transferred over that way. Okay, so it's now 9:39, or 3:59, I can't read. 3:59, so I'm going to go ahead and check back with this glucose solution. So with this glucose solution, it's going to be hot. It's in a test tube in hot water. This is called a test tube holder. You squeeze it, it opens. Okay, so I'm going to squeeze it, except my plastics. Okay, and I'm putting it towards the top. Notice I'm still picking it up away from me. And if you look and you see a little precipitate at the bottom, probably a gold color, you have simple sugars present. Simple sugars are the ones that are going to be cashed in, used up right away. They're not going to linger around. And so, like for instance, this one right here, it looks like it has a little bit of yellow precipitate in it, but you'll be able to tell from your batch a little bit easier. Now, after each of your tests, you're going to want to clean out your test tubes. If there is a precipitate in your one that was in the water bath, you will have to use a test tube brush also to maybe scrape it out. The important thing is, and it says it in your lab, please do not put a hot test tube in the water. You must wait for the test tube to cool down before you can rinse it out, or else the glass will actually break. So just put it in your test tube rack while you're waiting. When you're all finished and cleaned up your area, you can go back over to one of the other lab tables or desks to work with your group, and that should be the end of it. Just check with Mrs. Shannon, okay? Hopefully this helps, and good luck. Make sure you complete the egg puzzle before you go on to the lab. Thank you.